Good afternoon, good morning if you are joining us from the States, from America. My name is Krzysztof Stawujak. I'm VP Sales at Solvita, and I'm very happy to be your host today. We're excited to have you here as we'll explore practical applications of mass spectrometry-based proteomics in drug discovery and development. We'll begin with a presentation from our speaker, Dr. Alexander Moisa, who will discuss the scientific and technical aspects of proteomics workflows. After the talk, we'll of course move to the Q&A session. So please feel free to submit any questions that you may have at any time during the webinar using the chat window. Before we begin, I would like to introduce today's speaker, Dr. Alexander Moisa. Is a biochemist with over 10 years of experience in mass spectrometry-based proteomics and metabolomics. He specializes in the LC-MS method development and data analysis and support drug discovery and development projects across pharma. At Solvita, he works as an analytical expert, leading complex studies and delivering results to our clients. Before I hand over to Alexander, one more thing, please let me briefly introduce Solvita to those of you who may not know us yet. We're an integrated service provider supporting both drug discovery and drug development. Our services span a wide range of areas from medicinal chemistry, DMPK, pharmacology, translational studies, to analytical services, formulation, biologics, characterization, and stability studies. We are proud to collaborate with a number of pharmaceutical and biotech partners to advance life-saving therapies, and proteomics is, needless to say, one of the technologies that we use to support our activities. So, without any further ado, Alexander, please go ahead.
Hi, Krzysztof. Hi, everyone. Thank you very much for the introduction. Okay, we can start. Today, presentation, my aim is to provide some background about proteomics. What is the proteomics? How we do it? Why we do it? I aim to give some understanding how proteomic studies may accelerate drug discovery, drug development processes. While I won't go into deep technical details, I would like to give you some understanding of how mass spectrometry works, because it's important for understanding what kind of data can be obtained in the experiment. However, first, what is a proteome? The proteome refers to the complex set of proteins expressed in the cell, tissue, or whole organism. Unlike the genome, which is relatively constant, the proteome is dynamic. It changes in response to environmental conditions and cellular processes. The proteome is much larger than the number of genes. A single gene can produce multiple proteins through the alternative splicing. Additionally, proteins often undergo post-translation modification, which further increase complexity. At every stage, synthesis, processing, and regulation, the number of distinct protein forms can grow. So how big is the human proteome? The human genome contains around 19,000 protein-coding genes. The Human Protein Project has detected around 18,000 proteins. The UniProt knowledge base lists around 20,000 reviewed human proteins, plus around 22,000 of alternative splice isoform, totaling over 42 distinct sequences. And we would like to identify and quantify as much as possible protein from this very, very long list during our experiment in drug discovery and drug development studies. Proteomics is the science and methodology of study of the proteome. The emergence of mass spectrometry-based proteomics was driven by several key scientific and technological advances. Advancement in genomic, in particular the sequences of DNA, allowed the prediction of protein sequences. Advances in mass spectrometry instrumentation, for example, electrospray ionization, high-resolution mass spectrometry, increasing sensitivity and acquisition speed, enable a high throughput identification and quantification of proteins. So together, genomic information and mass spectrometry have transformed proteomics into a powerful tool for understanding biology, especially in the constant context of disease mechanisms and drug discovery. Mass spectrometry is a powerful and widely used tool in proteomics. However, it's not only technique for studying the proteome. Other important approach includes protein microarrays and two-dimensional SDS-PAGE. In fact, SDS-PAGE was widely used for proteome analysis before mass spectrometry became the dominant method. As is usually the case, all methods have their advantages and disadvantages that I tried to list in this table. However, mass spectrometry offers a unique combination of strengths: high sensitivity, the ability to identify post-translation modification and their localization, the capability to distinguish between different protein isoforms. These capabilities make mass spectrometry an irreplaceable tool for comprehensive and in-depth proteome analysis. Since proteins play a key role in many biological functions, and their dysregulation is often linked to various diseases and disorders. Therefore, proteins are critical targets in drug discovery. Protein can also serve as biomarkers for diagnosis, prognosis, and treatment monitoring. Therefore, the high throughput identification and quantification of thousands of proteins are invaluable for drug discovery project. Target discovery, target validation, investigation of mechanism of action, of target analysis, and many other techniques require proteome profiling. These approaches may have different in vivo and in vitro components, but they share a common reliance on mass spectrometry-based protein identification and quantification. Since proteins can serve as a drug, mass spectrometry-based proteomics plays also an important role at all steps of the development and manufacturing biologics. Key implications include characterization of therapeutics protein, including primary structure investigation, identification post-translation modification during the discovery and development stages. Also, this characterization include quality control during preparation, biologic investigation, comparability, host cell protein profiling, and investigation degradation products in the later stage of development of and manufacturing biologics. How does a typically mass spectrometry-based experiment look? A proteomics experiment contains multiple complicated steps of sample preparation, mass spectrometry data acquisition, and data analysis. Generally, the workflow can be divided into three main stages: experimental design, sample preparation, mass spectrometry detection, and processing of mass spectrometry data. Since there are many different approaches for the sample preparation, mass spectrometry detection, and data analysis, it is essential to plan the experimental design at the beginning. A well-planned proteomic experiment is crucial for obtaining reliable and biologically meaningful results. During the discussion of the experimental design, first of all, we need to answer the question: is this experiment hypothesis-driven or discovery-based? If a hypothesis-driven approach is required, meaning that we are focused on a specific set of protein, then a targeted mass spectrometry method should be selected. Is the goal to identify and quantify as many proteins as possible without a prior assumption, as we have in a discovery-based experiment, then an untargeted method should be selected. We also need to calculate in advance how many samples and replicate will be included in the study. Based on this, we should select an appropriate peptide separation and acquisition strategy. For discovery-based experiment, also very important post-processing and data analysis, because we need to plan how many samples, replicates, time points for this experiment, because this data is requirement for obtaining a reliable data at the final stage of the experiment. Sample preparation for the proteomics experiment consists of several main stages. It includes protein extraction. For some cases, protein extraction may include protein enrichment for low abundant protein or depletion, high abundant protein. After that, we have a protein digestion. And after digestion, the resulting peptide mixtures can be analyzed directly by LCMS or can be additionally fractionated to increase the depth of protein coverage. There are two main types for this moment of hybrid instruments for proteomics studies. This is a QTOF, which combine a quadrupole and time of flight detector, and Orbitrap-based mass spectrometers, which combine a quadrupole and Orbitrap detector. Despite the differences between these systems, all mass spectrometers share a common structure. Each typically includes an ion source for sample ionization, a region for ion acceleration and focusing, a component for ion selection for detection of fragmentation, and a final stage for ion separation and detection. Protein identification in proteomics experiment using mass spectrometry is based on peptide identification. Peptides are identified based on their mass and fragmentation spectra combination that is nearly unique for each peptide. Therefore, mass spectrometry data typically include mass spectrometry spectra of peptide ion and their corresponding fragmentation spectra. Here you might ask me, why do we need to perform protein digestion? Why can we analyze intact protein directly? We typically do not use intact protein because they have a significantly higher number of charged states compared to peptides. This makes the mass spectrometry spectra of protein mixture very complex and difficult to interpret. In addition, the fragmentation spectra of intact protein also more complicated than those for peptide, making a reliable identification harder. In this slide, you can see an example of mass spectrometry spectra for the peptide and fragmentation spectra for peptide and protein. Fragmentation of intact protein, also known as a top-down proteomics, is used for detailed protein characterization in case of biologic, but not for complex samples such as those obtained from in vivo or in vitro experiment. In those cases, a bottom-up approach, which involves protein digestion, is a standard approach for the experiments. During processing of mass spectrometry data, the comparison of experimental peptide spectra with the theoretical allows us to identify the peptides. Based on the list of identified peptides, we receive a list of identified proteins. In the final list of identified protein, we usually have terms like a master or leading proteins. This term indicates that the protein has the largest number of unique peptides. This is important because it is possible, and it's often there is such situation when the same peptide may belong to multiple proteins. Since protein quantification is performed based on a peptide quantification, a shared peptides cannot be used for it. Only unique or rather peptides are used for quantification. And for reliable quantification, a protein should have at least 2 unique or rather peptides. Therefore, other proteins should be excluded from the quantification. There are two main strategies for this moment for data collection during mass spectrometry analysis. Main strategies, this is data-dependent acquisition and data-independent acquisition. Data-dependent acquisition involves fragmenting the most intensive peptide ions selected during each cycle of analysis. Data-independent acquisition fragments all ions with a selected M of the window without depending on intensity. At the beginning of mass spectrometry-based proteomics, data-dependent acquisition was the primary approach for data collection. However, data-dependent acquisition has several limitations during its reliance on ion intensity and inability to capture all ions. Today, data-independent acquisition is widely used, offering advantages such as fewer missing data and better reproducibility. While data-independent acquisition requires more complex data analysis due to the multiple fragmentation spectra in a single acquisition, current technologies allow for efficient processing of this data. Both methods are widely used for the label-free relative quantification of proteins. There is another popular approach is the using a chemical labeling of peptides with isobaric tags. This method increases the throughput of analysis by pulling several samples together before analysis. In this experiment, acquisition is performed using data-dependent acquisition, and quantification is based on the intensity of reporter ions that correspond to each specific sample. The intensity of report ions reflects the relative abundance of the target molecule, allowing for quantitative comparison between samples collected in the experiment. To summarize the section of mass spectrometry data collection, I have prepared a table of comparison different quantitative approach in mass spectrometry-based proteomics. So all these approaches we can split into two main groups. One suitable for absolute quantification, another for relative quantification. For relative quantification, there can be also two approaches, label-free and labeling. Labeling allows to increase the throughput. However, there is a limitation in the number of samples and price of the reagent. Labeling also allows to increase the depth of protein coverage by the fractionation of the pooled samples. After processing of mass spectrometry data, we obtain a list of identified protein, each with an associated score. In the peptide information for each protein, we can also find any identified post-translation modification. If a quantitative experiment was performed, the result will include values related to protein abundance. However, the representing proteomics data solely as a table is not always convenient. Especially when we have several sample groups that contain multiple samples. Some of the common methods for visualization of this data I will show in the next slide. Also, there are many ways of visualization proteomics data. Here we have four of them, and this is the most often used method for visualization. First is the volcano plot, shows both the magnitude and significance of protein expression differences between two groups of samples, highlighting significantly up or down regulated protein. Venn diagram illustrates shared and unique differentially expressed protein across multiple groups, helping to identify common and condition-specific proteins. A heat map displays protein expression across samples using color gradients with clustering to reveal similar expression patterns among protein and samples. And also very often use a principal component analysis. Principal component analysis allows reduced data complexity and highlight differences and trends across group, making an overall data structure easier to interpret. To demonstrate the versatility of proteomics for drug discovery, I collected some results from different experiments. The first one result is changes in protein abundance in cells treated with one compound. In this experiment, we use label-free quantification with data-independent acquisition. This approach allows us to identify around 1,500 protein. And this analysis reveals the presence of some down-regulated proteins. Another type of experiment may be a chemical pull-down combined with mass spectrometry. In this approach, we utilized TMT labeling, and we identified around 1,000 protein. This protein were relatively quantified, and around 200 protein will identify showing specific binding to the compound in this experiment. Also, TMT labeling can be subject to identify and analyze different type of samples. In the next experiment, we using proteomic profiling of serum, cells, lizard, and tumor intestinal fluids. And we use TMT based quantification, and also we include high pH fractionation that allows us to identify around 7,000 of protein. And you can see result clusterization of this experiment, hierarchical clusterization of protein abundance revel sample-specific and tumor-specific expression patterns. And the last one example, this is a limited proteolysis coupled with mass spectrometry for identification of binding site in cell lizard for the protein. A limited proteolysis connected with mass spectrometry we use to use for the confirmation of a binding site. And in this experiment result, you can see that we found differences in peptide fingerprinting for the samples treated and non-treated by the compound. Another important example of using proteomic approach, this is host cell protein profiling for drug development area. Mass spectrometry-based host cell protein profiling allows identification of individual protein, including a low abundance protein and also immune invisible protein. This method enables precise tracking of process impurities across different purification steps, support risk assessment, and ensure compliance with the regulatory standard. Mass spectrometry-based HCP profiling is essential for providing product safety, purity, and consistency in the development of therapeutics proteins. For this presentation, we also collected the most frequently question, and I select the top 3 question, and this is the number of identified protein, and the question regarding the accepted protein wasn't identified, and the question regarding missings in the mass spectrometry data. So the first and the most important question, this is a number of identification. And the number of identified protein highly depend on the type of the sample and sample preparation strategies that it was used for the experiment. However, there is some common features for it. For example, for plasma or serum sample, around 1,000 protein can be typically detected. This relative low number is due to the presence of highly abundant proteins which can mask the detection of lower abundant proteins. Depletion of highly abundant proteins can significantly increase the number of detectable proteins, for example, up to 2,000-3,000. For tissue samples, around 2,000 and 3,000 protein can be identified. For cell lysate samples, this number can exceed 3,000 or 4,000. However, the results for the number of identification protein highly depend on the type of tissue and cells used in the experiment. Sample preparation with extensive fractionation, for example, offline high pH fractionation, can increase number of detectable proteins. However, it reduces the overall throughput of the analysis. Another very important question is this is a question when the protein wasn't identified. There are many possible reasons for it, for example, such as low abundance of the protein, poor solubility, or inefficient digestion of the protein. However, there is a defined set of the proteins that must be identified. It is better to plan a target mass spectrometry method and check relevant data for this protein in different available proteomics repositories to check is it possible to detect and quantify this protein using mass spectrometry-based approach. And the last one in our today's presentation question is missingness. Missing data is a common issue in proteomic studies, and also there are different reasons for missing data. It can be caused by the stochastic nature of the data-dependent acquisition, or low abundance peptides, or incomplete detection across replicates. Additionally, peptide ion suppression or variability of sample preparation can contribute to missing values. And also, a different condition of instrument has also significant impact on the missing values. There are different ways to handle with missing data. One is filtering of this data, and another is imputation. The choice of method depends on the amount of missing data and design of the experiment. Filtering is often used when missing values are extensive and random, while imputation can be applied when the missingness is minimal or systematic. And to sum up our presentation, I would like to highlight some benefit of mass spectrometry-based proteomics. So the collection of comprehensive information is advantageous in any field of drug discovery or drug development processes, as it facilitates a better decision-making, problem-solving, and strategic planning. In addition, high-quality data is crucial for the effective implementation of machine learning algorithm, artificial intelligence in drug discovery and drug development. Mass spectrometry-based proteomics provide a unique capability for in-depth characterization of protein in complex samples, increasing the likelihood of identifying meaningful biological signals through the drug discovery process and enhancing the safety of proteins through the comprehensive characterization biologics.
Thank you, Alexander. We already have some questions, so I think that we can move forward to the Q&A session. If you haven't already, please feel free to ask questions. Just type them in the chat window, and we'll be happy to answer that. But the first question that I see is coming from Gabriela, and it's about sensitivity. How sensitive is the DMS-based proteomics? Can you detect low abundance proteins?
Okay. The Cation-8 mass spectrometry is now is highly sensitive and allows to identify protein with a concentration low nanomolar or nanogram or picogram concentration. Yes, but however, the success of identification and quantification of low abundant protein highly depends on type of the samples. For example, if the sample contain a highly abundant proteins that can mask low abundant protein, and also depend on the sample preparation strategy. As I told during the presentation, fractionation of peptide mixture after digestion allows to make the protein mixture less complex, and mass spectrometry analysis of obtained peptide fractions allows to identify and quantify more protein. However, this approach reduce the throughput of the analysis. So, but you can see from the data in the presentation that, for example, for cell lizard sample or cell pellet sample, it's possible to identify up to 10-12,000 of the protein. So it's a very, very high number of protein.
Thank you. There is another question also from Gabriela, and that's about the delivery of data. How is the data delivered? Do you provide only raw files and also, or also analyzed results?
Okay, thank you for this question. We can provide also raw data is it required for the client, because sometimes clients would like to perform their own processing of mass spectrometry data. We also can perform mass spectrometry data in data processing in our site and provide a list of identified proteins. We also can perform post processing of mass spectrometry data, and we discuss what we should do with the mass spectrometry data during proposal preparation and discussing this experimental design.
Thank you. There is another question coming from Javier, and it's about normalization of results. How do you normalize the results between replicates, and how do you quantify proteins?
No, regarding quantification proteins, there are different approach. For example, if we are talking about label-free quantification, we quantify protein based on their intensity, based on intensity of the peptides ions, and we can normalize data based on the spiked internal standard. There are commercially available internal standard for such experiment. It contains peptides that cannot be in, for example, in the human sample, and we can use this standard for normalization. Also, there is another approach. We can normalize all signal based on a total ion intensity from the whole set of the protein. And regarding for TMT labeling, there is also the similar approach when we use normalization of the total protein contained in the sample, and it allows to normalize samples. But usually we perform analysis with normalization and without normalization to check is there any influence of normalization on our data.
Thank you. There is one more question coming from Tomasz about internal standards. Are you using internal standards for label-free measurements?
Yes, we use it, yeah, this internal standard for label-free measurements.
And the last one I see is coming from Artem, and it's about artificial intelligence. Do you think that AI or machine learning will play a major role in interpreting complex proteomics data in future drug discovery?
From my view, artificial intelligence already play an important role in data analysis for mass spectrometry data. For example, there are approaches for the protein identification where it uses the predicted fragmentation spectra using artificial intelligence and machine learning algorithm. So we don't need to use a database that contain, for example, fragmentation spectra for the peptide. You can use this data. So I think artificial intelligence and machine learning algorithm will play a very important role, and this role will increase in proteomics experiment, in other experiments in drug discovery.
Thank you. I see that someone is still typing the question. Artem, I genuinely appreciate the clarity and the passion you brought to this topic. Thank you. That's very kind. And a question from Javier. How reproducible are results from biological replicates with high fractionation process? Question.
Yeah, this is a very difficult question. It's difficult to answer your question, but this is a very important question. I think it's also highly depends on the type of biological experiment. What type of sample we have? Of course, the fractionation increase the error between replicates, but it allows to identify more proteins. But in general, the biological replicates is something like a very, very important things, but it produces a much more error for the calculation.
So there is a definitive trade-off. I still see that someone is typing a question. So maybe we'll wait just one moment. There's a thumb up from Javier. So
thank you,
I think that the answer is the reply is well received. We see another question from Ramesh. Just a moment. What were the major technical or analytical challenges that you face in this project?
So it's a difficult question. Of what type of project did you mean? However, the main challenge in project this is reproducibility. So if you have a long-term project and you have a lot of samples and you have to analyze in this sample in different batches and in different days or weeks, this is a very difficult point to provide a good reproducibility and consistently with data between these samples.
Thank you. And maybe I will ask a question. Are there any matrices, biological matrices, that you find typically the most difficult to work with as compared, for example?
The most I think the most difficult matrices is plasma or serum samples, because it contains a lot of high abundant proteins that can mask a low abundant protein, and this very difficult matrices. For example, if you need to identify some low abundant proteins, you have to perform protein depletion or enrichment. If you, for example, have some target protein and it significantly increase the difficulty of the protocol and increase the challenge in this experiment.
Okay. There is a thank you note from Ramesh, and another question statement from Javier. If I'm not wrong, you said that the DDA was more reproducible than the DA.
No. Data independent acquisition more reproducible. Maybe I made a mistake during talking. Yeah, of course, data independent acquisition is more reproducible.
But one way or another, we can submit it, because Javier is asking about the paper. So we are always happy to follow up with the questions. Since that was, I think, clarified. But if you have any other questions, please feel free to ask. And if not, we'll be happy to follow up by email, ideally. I don't see any other questions. I also don't see any. Oh, someone is writing as well. There's a question from Ignacy. Do you offer target engagement and downstream PD biomarkers development?
Yeah, we can support such studies. We have collaboration with our biology department and can support different in vitro experiment, and also we have capability for in vivo experiment, I guess, and we can support different type of experiment, including targeting and engagement. And regarding biomarker, also we can support this experiment.
Thank you. And I think that it's also for the off targets, right? So that
yeah. Yeah, for off target also we can support it. Yeah, in case of like I told during this this presentation, different type of target discovery experiment may have a different in vivo and in vitro part, but finally, for example, if we are talking of mass spectrometry-based proteomics, this is a common type of experiment for different method, and drug discovery project. So we can support any drug discovery project that is related with needed of protein identification, protein characterization, and protein quantification.
Thank you. There is a note from Javier saying that this confusion was made in the table, in the presentation. We'll certainly look into that.
Yeah, we will check it.
As anyone, we're not free from making typos, right? So sorry for the inconveniences. Next time will be better, right?
But it's good too that experienced person
is actually reading the table,
yeah,
right? So that's good. Thanks. I don't see any other questions. And with that, I think that we can safely conclude. Thank you for the presentation. Thank you for all the answers. There's another one, so maybe. There's a thank you note from Gabriela. And another question is probably coming. So there is a thank you note from Ramesh, from Gabriela. That's actually very kind. And there is a question from Javier. Do you have already tried ReadyBeats technology for proteome quantification?
No, we didn't try this technology.
Oh, sorry.
Yeah, sorry. But if you have any question regarding, for example, organization such experiment, feel free to write, send us email, and we will contact with you.
So we certainly stay available for answering questions. Thank you. So with this, I guess that we can conclude. Thank you very much for being here, for listening to Alexander. We're very happy to see you. If you still have questions, or maybe there is something that you'd like to have clarified, please feel free to reach out to Alexander. We'll be, I guess, also posting this webinar somewhere online. And yeah, we hope to see you again, maybe on another occasion, another webinar. So hopefully.
Thank you very much for the participation and the questions.
Thank you very much. Take care.
Okay.